Well, hello and welcome to part two of this introductory discussion on cell anatomy and physiology, looking at mainly prokaryotic cell anatomy and physiology and a little bit of eukaryotic. I want to apologize for not properly ending part one. I ended it very abruptly because I just kept going. I wanted to finish earlier, and I realized as I was going on, I thought I didn't want to go this far. So that's why part one ended so abruptly, but I just went on too long. So let's pick up with part two. We had been talking about gram-positive, looking at the gram-positive characteristics and comparing gram-positive with gram-negative. Now let's look at gram-negative, specifically gram-negative outer membrane. So the gram-negative bacteria, they have this outer membrane that is complex, way more complex than peptidoglycan is of the gram-positive bacteria. So they have an outer membrane that's made of lipopolysaccharides, lipoproteins, and phospholipids. And there is this thin space called the periplasm. Well, in gram-negative, the periplasm, there's more of it than there is in gram-positive. What does this outer membrane do for the cell? It protects them from phagocytes, so white blood cells, macrophages, complement proteins, which are useful in puncturing holes in cell walls, antibiotics, and enzymes, which can degrade peptidoglycan very easily, like the enzyme lysozyme. Well, gram-negative have the benefit of this outer membrane, and so they're resistant to these things. They have an O polysaccharide antigen, which is useful in distinguishing variants within a species. So if you remember the E. coli example, E. coli O157H7, the O polysaccharide is in the outer membrane, and we can use that in not just E. coli, but other gram-negative bacteria as well. It contains lipid A, and this is the endotoxin, which we'll talk about later when we talk about the toxicity within bacteria and what is a toxin and what do they do. An endotoxin comes from within the cell. So this is embedded within the outer membrane, and it's called lipid A, which causes fever and or shock when in the host. So shock is uncontrollable decrease, severe decrease, and sudden decrease of blood pressure. And porins, which are proteins, they form channels through the membrane to allow things to enter and exit the gram-negative cell. So here, what it looks like up close, you have the outer membrane, the thin layer of peptidoglycan, and then the inner cell membrane. And as you can see, and this picture doesn't do it justice, but the outer membrane is complex, way more complex than peptidoglycan, the outer layer for the gram-positive cells. So we had in the last chapter, in the chapter on microscopy, we had discussed preparation for specimens for viewing under a microscope. And one of the staining mechanisms was the gram stain. And I'd said we'd look at that a little more closely. So the gram stain mechanism, what happens is you add crystal violet, that's your stain, and then you add a mordant called iodine, and those two join together and they form these crystal violet iodine crystals within the cell. And then you apply alcohol and let it sit on your smear for a few seconds. Well, what does that alcohol do to gram-positive? It dehydrates the peptidoglycan, making it impermeable to the CVI crystals. So now the crystals cannot get out of the cell, and so the cells appear purple as a result of the alcohol dehydrating it and just simply the large size of the crystal violet iodine crystals when they chemically join together. With gram-negative, you have that outer membrane made of lipids. The alcohol dissolves the outer membrane, leaves holes in the peptidoglycan as well, and the CVI crystals, then they wash out and the cell is colorless. Then you add the counterstained safranin, which is red, and those cells now appear red under the microscope. Now you also add safranin to gram-positive, but the crystal violet is so intensely purple that the red, you can't even see it. It doesn't make any difference to the violetness of the cell. So gram-positives remain purple, gram-negatives, they're red from the safranin. How do we damage the cell wall? What are ways that damage can be done to it? Well, the enzyme lysozyme, which is an enzyme that we produce in our bodies naturally and pretty much all of our bodily fluids, digests disaccharides and peptidoglycan. So the disaccharides are the NAG and the NAM. So break down those disaccharides, the raft can then fall apart. It kind of breaks it down, dissolves it. Lysozyme is produced in tears, saliva, and mucus. Penicillin, the antibiotic penicillin, inhibits the peptide bridges. So what happens, if you remember the log raft analogy that I used, those peptide bridges are like ropes holding the logs together. And if the ropes are disintegrated or broken apart, then the raft falls apart. The peptidoglycan falls apart. And then you're left with, in some cases, a wall-less cell called a protoplast, and it's susceptible to bursting, osmotic lysis. Protoplast can happen naturally through the use of, I guess it's not really natural for the bacteria to not have a wall, but if we use penicillin, they can become wall-less that way, or we can use protoplasts for research, where the cell wall is intentionally removed, and then we can do research on these cells. Now we have a picture here, a micrograph. The one on the right is an electron micrograph of a bacterial cell. You notice it's got an outer membrane, the peptidoglycan, and then the inner membrane, the cell membrane. Now, can you tell by looking at this picture, is this bacterial cell gram-positive or is it gram-negative? What do you think? It is gram-negative because of the outer membrane. So you're looking at a gram-negative cell wall here under an electron microscope. Let's look at the plasma membrane. It's a phospholipid bilayer. So it's got those yellow spherical structures in that picture. Those are the phosphate groups, and then the squiggly tails are the lipids. And it's a bilayer. There are 2 layers of them. So if you can look at it and label them with their phosphates and lipids, starting at the top, you'd have phosphates, then lipids, then phosphates, and then you're inside the cell. There are proteins that are throughout the cell. They're called peripheral proteins that are on the surface, and the ones that are embedded in the cell membrane are integral proteins, and the ones that go the whole way through, those are called transmembrane proteins. Those terms are used interchangeably. Sometimes they'll call, like you see in this picture, an integral protein. You could be more specific and call it a transmembrane protein with a pore that runs through the middle that enables the passage and transport of materials into and out of the cell. Now there's this term that we use called the fluid mosaic model. Fluid means the membrane it's referring to its ability to move around. It's very it's about as viscous as olive oil. And so things can move and squeeze their way through the plasma membrane rather easily. The plasma membrane itself, the phospholipids, are constantly in motion. The proteins are squishing in between them and moving around so that they can carry out their functions in different areas of the cell. It's called a mosaic because there are a mosaic of compounds. There are a number of different compounds which are embedded within it. So fluid meaning it's always in motion, things are moving through it. This enables the transport of materials easily into and out of the cell. And then it's a mosaic, so there's a variety of compounds that are within it. The plasma membrane is selectively permeable, as opposed to semi-permeable, which is a term that's sometimes used, and I think that's incorrect. Semi is saying that if it's small enough to get through the membrane, it'll get through. But that's not the case with cells because there are ions, charged particles, that are incredibly small. They could easily pass through the phospholipids, but they can't because they have a charge. The lipids resist anything that's polar or certainly charged. So selective permeability, or selectively permeable, says that the membrane can let some things in, but not all things. And it can be small enough to get through, but in some cases it rejects those things for one reason or another. There are enzymes within the plasma membrane to produce ATP, which is the energy molecule needed for cells. Remember, bacteria do not have organelles, and so they don't have mitochondria, so they don't carry out cellular respiration through mitochondria. Instead, they have to rely on enzymes in their plasma membrane to do the ATP production process. If they are photosynthetic bacteria, their pigments are found in infoldings in the cell membrane called chromatophores or thylakoids. So they don't have chloroplasts, so they don't carry out photosynthesis through those organelles. Damage to the membrane by alcohols, detergents, and antibiotics can cause leakage of the cell contents into the extracellular environment. That's going to lead to cell death. And so the membrane becomes the #1 target for our chemical compounds and our antibiotics. If we can take apart the membrane, the cell will lyse or it will burst. And my micro students, you can when you're on the actual PowerPoint, you can click on this hyperlink and you can watch a pretty good TED Ed video on traits of the cell membrane. Good animations. It'll help you understand a little better. Let's look at ways that materials can move into and out of the cell. We'll start with passive forms of transport. Passive meaning no energy required. There's simple diffusion, which is a movement of a solute or a particle from an area of high concentration to low concentration. So it's just moving with the flow or moving with the concentration gradient. And then there's facilitative diffusion, in which the solute combines with a transporter protein in the membrane. So to facilitate means to help. So there's a helper protein that's going to enable the passive diffusion of the solute from inside the cell to the outside or vice versa. And neither of these processes requires energy. So we call them passive processes. Here's a very simplistic picture of facilitative diffusion. You've got particles getting caught up in a transmembrane protein, and then they're passed through the cell to the inside of the cell. Many times these transporter proteins are very specific to their transported substance. What I mean by that is they only allow one particular compound, if it's a protein or whatever, to pass through it and nothing else. So they're highly specific, highly selective. Some transporter proteins aren't. They allow a variety of materials to get through as well. But for the most part, they're pretty specific as to what they allow to pass through them. Osmosis is just basically the diffusion of water. The simplest definition would be the diffusion of water. The movement of water across a selectively permeable membrane from high water concentration to low water concentration. And so what you see in this picture here is you have a glass tube with what they label as a cellophane sac that's down inside the water, and it's got solute in it, in this case sucrose or sugar. And what happens is there's a higher concentration of water on the outside, meaning there's a lower concentration of solute on the outside. So water will naturally diffuse through the cellophane sac inside of it, and it'll cause the water to rise up in the glass tube. So this is a simple experiment that proves water will move from a low solute concentration to a high solute concentration, or if you look at it another way, from a high water concentration to a low water concentration. And osmotic pressure is the pressure that's needed to stop the movement of water across the membrane. Eventually it'll reach equilibrium and water will move in and out at an equal rate. So speaking of osmosis, let's look at the different types of solutions. There are three types of cellular solutions based on solute concentration. There's isotonic, hypotonic, and hypertonic. The prefix iso means equal, and the suffix tonic comes from a Greek word that means strength, so equal strength. There is no net movement of water here. This is where the concentration of solute on the outside of the cell is equal to the concentration of solute on the inside of the cell. So because everything is equal now, water's moving in and out at an equal rate. And so this would be in a state of what we would call equilibrium. The next one, that's easy to understand. The next one, hypotonic. Hypo means less than, so less strength. In this case, there is a lower concentration of solute on the outside. Those little dots, those represent solute, like sugar or salt or whatever. And so water will move from the outside of the cell, where there's a low solute, to the inside of the cell, where there is a high solute concentration, in order to create equilibrium. And if this environment is overly hypotonic, it'll cause the cell to swell up, but bacteria have cell walls which prevent them from bursting. So if we can damage the cell wall, then they'll burst, they'll lyse. And that's called osmotic lysis or cytolysis. Hypertonic, hyper means greater, greater strength. In this case, there's a greater concentration of solute on the outside of the cell. So there's a whole bunch of salt or sugar on the outside of the cell, which causes the water to osmose from the inside of the cell to the outside of the cell, to the outer extracellular environment. This causes the cytoplasm and everything to kind of shrink and shrivel up, and this can lead to the death of a cell as well. That's called plasmolysis. And we take advantage of this, like for example with salting foods. Creates a hypertonic environment, bacteria land on the salted food, water osmoses out of them, and they die. So this was a method of food preservation, and of course still can be, but we have refrigerators now, so we don't bury our food in salt like people did couple hundred years ago. So isotonic, hypotonic, hypertonic. Let's look at ones that are active. Forms of movement that are active or require energy. The movement of a solute from regions of low solute concentration to regions of high. So going where there's a low quantity of that solute, the solute moves from where there isn't much of it to where there is a lot of it. So it's going against the concentration gradient. In order to do that, it requires a transporter protein, and ATP is necessary as well to force the solute to areas where it's already highly concentrated. ATP is the energy molecule that's used in order to make active transport occur. In bacteria, there is another form of active transport called group translocation, where a substance, which is usually a sugar, is chemically altered while being actively transported across the membrane. This requires a protein as well, and another energy compound called PEP. I guess that's pretty easy. It's peppy. It gives it energy. It enables it to cross the membrane. And this, again, is only found in prokaryotes. Eukaryotic cells do not carry out group translocation. The cytoplasm. The cytoplasm of a bacterial cell is about 80% water. Contains proteins, carbs, lipids, charged particles, and ribosomes. And in the micrograph on the right, all that granular appearance, that's because of the ribosomes that are in it. There are thousands of ribosomes in bacterial cells. But notice, no organelles. And its water content is higher than humans. Human average water content is around 65 to 70% on average. So they have a higher water concentration in their cytoplasm than many eukaryotic cells do. Remember, bacteria, prokaryotes do not have a nucleus, but they have an area where the DNA can be found, and that's called the nucleoid. And if a word has a suffix oid, it means it's like the prefix. So a nucleoid is like a nucleus, but it's not. There's no membrane surrounding the nucleic acid. And there are other segments of DNA called plasmids. These are circular pieces of DNA, and we use plasmids for all sorts of experimental purposes in genetic engineering and so forth. So nucleoid, no nucleus, but a region called the nucleoid. Ribosomes are, as I said earlier, complexes of RNA and protein. They're the sites of protein synthesis. A process called translation occurs there. They number in tens of thousands in the bacterial cell, and they give the cytoplasm a granular appearance. So ribosomes are non-negotiable. All forms of life have to have ribosomes because that's where the proteins get made. That's where the RNA is read. And so all forms of life have to have some way of reading those instructions and then making proteins. And that something is a ribosome. Inclusions are reserved deposits within the prokaryotic cytoplasm, so they don't have organelles. So they have to have something that replaces the function of organelles, and these are called inclusions, where they can hold things like they have energy reserves, phosphate reserves, and so forth. I'm not requiring my students to know these, but I just gave you a number of these different inclusions and what their purposes are. Just know that the inclusions, they're kind of like a replacement for the organelles. So the bacteria need these reserves. They need to store these things up because they don't have a way of manufacturing it. So they have inclusions instead. Endospores. Endospores are like hibernating cells. They're resting cells with very thick cell walls and additional layers on top of that. And they can stay in this state for thousands of years without dividing. No metabolic processes are going on. But you still have that cell wall, and you've got the nucleic acid that's still viable. They're very resistant to desiccation, which is totally drying them out. High temperatures, low temperatures, chemicals, radiation. And two groups of bacteria that are very good and commonly form endospores are the Bacillus group and the Clostridium group. And the term sporulation is the formation of endospores, and then they can return to an active state, and that's called germination. where they become, we say, a vegetative state, or a reproductive state where they become metabolically active and they start dividing. So whenever their conditions are right again and they're in the right environment, they can germinate and then start to multiply and divide again, even after being dormant for thousands of years. They're incredible. Here's a picture. It's not a great picture of an endospore, but there aren't too many good micrographs of endospores. So you can see the outline of the cell wall, and then that dark center oval mass, that's the endospore. Nucleic acids are still there, so all the instructions on how to be a bacterial cell are still there. And whenever it's in the right environment, that endospore can become active and they can start multiplying and dividing again. Well, I know where to stop this time. This is where I wanted to stop for part 2. So I hope you enjoyed part 2 and that it was of educational benefit to you. Stay tuned for part 3. This is a 3-part series. Part 3 will be looking at eukaryotic cells. Thank you for watching.